So in this lecture, we are going to discuss some very basics about the flow cytometry. So this lecture is not for the experts of the flow cytometry. So flow cytometry, you might have seen two terms which are very common, which one is the forward scatter and the second one is the side scatter. So forward scatter basically tell you about the size of the cell, and this one, the side scatter, basically tell you about the granularity of the cell. And why it is so? Because what happened, that suppose the light is traveling like this, okay, and suppose you pass one cell, this is suppose your cell. So this light is going to be diffracted in the different direction on the basis of size. So this is known as the forward scattering. Similarly here, when the light is hitting the cell, on the basis of its internal structure, it is going to be reflected or refracted. Okay, and this will tell you about the granularity of the cell. So these are the two basic things. Now you have seen here the two terms, which is the diffraction, then the reflection and the refraction. Here you can also see light refracted or reflected at the interface between the laser and the intracellular structure. So this is your laser and this is your intracellular structure, and the light is going to be reflected or refracted. So on the basis of that, side scatter plot can be generated. So what is diffraction, reflection and refraction? Diffraction happened in any direction. Okay, so whenever light is traveling, as I said here, light is traveling, and suppose a cell comes in the path of the light, it is going to be diffracted. Similarly, reflection, when the light hit the surface and bounces back. For example, light hit the surface and then bounces back. This is the reflection. And what happened in case of the refraction, light comes and then it bend or something like that. This is known as the refraction. When the light is passing from one medium to other medium of the different optical density or the different refractive index, light could be refracted. So on the basis of light diffraction, reflection or refraction, these plots are generated: forward scatter and side scatter. And how the signal, you can see that this is the signal which you can see. So whenever the light is hitting the where is my pen? So whenever the light is hitting the laser beam, and once the your particle is within the diameter of the light of beam of light, then the signal is going to increase. Once the particle passes outside of the diameter of the laser beam, the signal is going to decrease. Okay, so this is the thing. So here you can see at this point the particle is fully illuminated. Okay, when the particle is located in the center of the laser beam. Okay, as the particle flows out of the laser beam, then the intensity of the laser beam or of the signal is going to be decreased. Here you can see. So this is the maximum intensity when your particle is within the diameter of the laser beam. When it passes outside of the diameter of the laser beam, the intensity will decrease. So what are doublets? When the two cells stuck together, this is known as the doublets. So that is what we don't want in our experiment, because suppose These are basically not one cell. These are the two cells, but our machine is reading just like a one cell. So the accuracy of our experiment will decrease if we are counting the doublet cells in our experiment. Here you can see when the two cells are stuck together, there is no change in the height, but there is a change in the area. Whenever you are doing removing the doublets, you need to plot FSCA or FSCW or H. You can also take SSCA or SSCW or H. Okay, so area is going to be increased. So you will be getting something like this, and there will be some cells here because this is the higher area. So this doublet you need to remove. You need to just gate your cells, these cells. Okay, so area of the cells will increase if there are doublets. Now coming to the compensation, what is compensation? That is the spillover between emission spectrum of two different dyes. So suppose this is the FITC dye and the emission spectrum is 525 to 50, and this is the PE dye and its emission spectrum is 585 to 40. But in here you can see this is the small portion that is coming. The PI is the emission spectrum of the PI, which is this one. The small portion, the blue part, is coming in the emission spectrum of FITC. So this is known as the spillover of PE to FITC. Similarly, in case of the PE, you can see the spectrum, emission spectrum of the PE is 585 to 40, and the FITC is going, its emission spectrum is spilling in this emission spectrum. So this is the region which is known as the spillover. For this, there is a small spillover of PE into FITC, but there is a large spillover of the FITC to PE, because you can see this is the emission spectrum of the FITC, and this is the emission spectrum which is going to be counted by the machine, by the detector, and this particular portion is coming within this 585 to 40 region. This is known as the spillover between emission spectrum of two different dyes, and that is what we don't want, the spillover. Okay, so for that we are basically doing the compensation. So whenever you are doing the compensation, it is very essential you do the compensation, you set the voltage first of your machine. For the single strain dye, for example, suppose this is the FITC dye, you take the cells, suppose this is your cells, this is the FITC labeled cells, you spike it with the unlabeled cells. So your positive population will come here, and your negative population will come here, and your positive population will come here in your facts when you will analyze the data. But what will happen if your negative population is here and your positive population is coming somewhere here? So you need to set the voltage in your experiment so that this will become like this. They should come linear to each other. Okay. This is known as a single cell compensation or setting the voltage of your experiment. So this is very essential. First you label your cells, spike your cells with the unlabeled cells. Your unlabeled cells should be here and your labeled cells should be here. But if suppose it is coming here, then you set the voltage so that they are coming here. So this is known as the setting of the voltage of your machine to accurately measure your data. Okay. Now what you can do, Now what you can do, but there may be 7 dyes also. Okay. Then you can do the compensation. You take the die, so there will be for all the 7 die, 1 die, then second die, only you should get the FTSE population. Okay. You should not get any other population while you are doing the compensation. For this is suppose you have 1 die, then your second die, okay, then your third die, like this. If suppose this population is coming somewhere here, then you need to again control the spill by just pressing the compensation tab. You can do that. That's not a very big aspect, but that is how the compensation is usually being done. For example, suppose you are performing an experiment in the lab uh, on 4 mice, let's say, okay, and you want to see the helper T cells, and you have treated, you have one mouse which is your control mouse and the other mouse which have been treated by the different drugs, and you want to see the expression of CD4, CD8, then granulocyte markers, CD45 are for B cells, CD11B for macrophages, okay, and these are labeled with this dye. Here you can see the macrophages. The marker of the macrophages is CD11B, which is labeled with the biotin. So biotin is going to react with the streptavidin. Okay, let me clear about biotin. So this is your biotin. This is going to bind to the streptavidin. One streptavidin can bind to the 4 biotin molecules, and then it can be labeled with the dye. So this is how biotin works. Okay. So in case of the biotin, you need to have the streptavidin. Okay. So these are the 5 cells you want to study. These are the markers and these are the dye which you are using in your experiment, let's say. So now you need to make the control cells for the compensate. You are saying, you will say these controls as the compensation controls. For example, you will take just mouse tissue just like that, unstained cells, or you will take the unstained cells or the mouse one, and cells from the mouse one, and labeled with the APC only. For example, here you can say the APC, then you have labeled the per-CP, then PE7, FTC, and this is PECI7 is a tandem die because they are made up of the 2 dyes. Okay, here what happened, the PE is transferring energy to the Psi7, and Psi7 is going to emit the spectrum at certain wavelength, but there is not 100% transfer of the energy, and therefore you will get some sort of spillage of PE into the PECY7 region, and that is why you need to have the PE control, and the biotin you need to take the streptavidin only. So these are the 7 compensation controls you will take. And how you will do that experiment, you will dilute the antibodies. Let's say if the dilution of the FTC antibodies you find in the literature or with your previous lab experience, it is 1:200. So you need to take 1 microliter of PBS and 199 microliter of this FTC only antibody. Take your cells and add in this tube. Like that you can do for the other tubes, like take the antibody dilution for the APC, add this in the unstained cells, like this for the other controls also. And how will you make the control tubes now? So for cells only you will take only PBS, there is no dye. In APC you will take only APC, and there are 4 steps, step 1 and step 4. There is a gap in between step 1 and step 4 because once you are adding the dye, you will incubate it for the certain period of time, centrifuge it, wash it, and then you will add another antibody if you want to add. So that is why this is the step 4. Per-CP only you will add only per-CP. PECY7 you will only add PECY7, but it has the PE, so you need to add the control of PE. So in this PE only you will be having the PE only and the PE streptavidin. If your streptavidin is labeled with the PE, then the FITC, in the FITC tube you will take FITC only, and abidin you will take only PE streptavidin. Okay, so like this you need to make the control tubes. For example, 50 microliter of the cells, 50 microliter of the PBS, and then in the step 4 you can take the 100 microliter. So total is going to be the 200 microliter, 50, 50 and 100. Okay. Now coming to your experimental group, suppose you have 4 mouse. One is your control mouse and other mouse which are treated with the different drugs. Then you will be adding the cocktail of all 7 antibodies. So how will you do that? If suppose you are making the total amount of 250 microliter and the dilution you know, then you can take 2.5 of this, which is 250 by 100, 2.5, 200 by 250, 250 by 200, which is 1.25, like this, and you can make it up to the 250 microliter. But here you can see you need to make the antibody cocktail for streptavidin also. Okay, so you need to make antibody for the antibody cocktail for the streptavidin as well, because biotin is separate. You can't mix it with these dyes. So for the streptavidin, suppose you have 4 mouse and there will be 2 controls here. You can see here, this is the PE streptavidin. For biotin you need to add the streptavidin, so that is why we are making the streptavidin cocktail. So 2 tubes there, and if suppose you have 4 mouse, so there are 6 tubes, so like that. And if the dilution is this much, so accordingly you can calculate the amount, which is 600 by 50, which is 12 microliter. Rest is going to be, which is 600 minus 12, which is going to be your PBS. Okay, so this is in the step 1 you will add the cocktail of these antibodies, which is 50 microliter, and in step 4 you will add from this cocktail you will add the 100 microliter of this streptavidin. So again 50, 50, 100. So that is how you can set up the experiment in the laboratory, and this I have discussed before. And annexin 5S is also done usually. So I just want to clarify, basically the phosphatidylserine serine is present on the inner membrane of the cells, but in case of the apoptotic cells it comes outwards. So annexin 5, if it is reacting with the phosphatidylserine, it means the cells are apoptotic. Just the principle of annexin 5. Similarly the PI, if the cells are dead, PI will react with it, while if the cells are alive, PI may not react. So this is something which I want to tell you about the setting up of the voltage. Here you can see you add the specimen and then you click here, activate this window. Once you activate this window, you will see all these tabs here. One tab is the properties, parameters. So what will we will do in the parameters? We will add the dyes which we want to add. After that we will acquire the sample by clicking the acquire data, and you will be getting something like that. If suppose this is coming here, if let's say this is coming some instead of here, if this portion is coming something like here, okay, then you need to set the voltage. And how you can set the voltage? There will be the button here. When you will click here, it will move from here to here, and that's how you set the voltage. Okay, once this is your instrument is set for the voltage, you can add the dyes. For example, you are adding the 7 colored dye, and then you go to the compensation tab, which is this one. Click on here and set up the spillover, which is this is known as spillover overlap. And here you can, if suppose, because there is a spilling of this antibody to FIT-C, so we will just move this spillover overlap and it will compensate everything, and you will get the data, and then you can use that data and compensate your samples in the Flozo or some other softwares. So this is all it for today's lecture. Thank you very much for listening me. We will meet in the next lecture soon.